Okay, so so I'm just going to give you a scientific update about where we stand on the academic side of the research part of it. There's a few companies involved in this, and I've got NDAs with them, so it's a little bit tough for me to talk about the industry side of it. And obviously, I'm interested mostly in where we are with the basic research. Although we do work with these companies, they're providing us with the recombinant human laminin, which I'll talk about in a second. So as everyone knows, laminin alpha-2 related congenital muscular dystrophy is caused by mutations in the laminin alpha-2 gene. It's either a complete or partial absence of laminin alpha-2, and this is critical for the formation of these heterotrimeric proteins, laminin 211 or 221, and each of these numbers designates the chain. So wherever there's an alpha-2, that's where you're missing that alpha chain. So patients with this disease don't form either of these heterotrimers. Laminin 211 is found around the muscle fibers, and then 221 is concentrated at the neuromuscular and myotendinous junctions. So for those patients that have complete absence, they're missing the laminin isoforms at the neuromuscular as well as the myotendinous junctions, as well as across the rest of the fiber. Characterized by severe progressive muscle weakness from birth, and obviously hypertonia, so straight from birth, there's an issue. Delayed motor milestones, peripheral neuropathy, and changes in brain white matter is detected by MRI. The muscle disease, as Carsten mentioned, occurs during embryonic development. So when you see the disease, you automatically know that during development, muscle development, there have been problems that have been occurring during that developmental phase too. Now, during development, we have laminin 111, which is present, and that seems to be able to compensate at least partially for the absence of 211 during embryonic development, but then it goes away. For some reason, and we don't understand why, we lose laminin 111 in our muscles, and now the predominant laminin is the adult form, laminin 211, which becomes predominant. And of course, patients with the mutation don't produce and switch to the adult form, and so there's none of these adult forms present. And currently, there's no treatment or cure, and everything is palliative. The treatment and care is palliative. So parents do an awesome job, and we understand. So in terms of the molecular mechanisms, I'm an alpha-7 beta-1 integrin person, so I always put that, but laminin also interacts with dystroglycan, which is part of the dystrophin glycoprotein complex. And in this case, you see that, see online if I can get my cursor here, in normal muscle, we have the adult forms of laminin. They bind to, in our case, the integrin and also dystroglycan. There's actually two binding sites, one for integrin and one for dystroglycan. And they're attached to in this case, the alpha-7 beta-1 integrin or dystroglycan. These are transmembrane linkage systems that link to the cell cytoskeleton. And so it forms a molecular glue that binds the outside of the cell to the inside of the cell, and therefore provides that structural integrity. So when you use your muscles, they're actually generating force against the matrix that's on the outside of the cell. So in laminin alpha-2, you're missing laminin, and actually what you have also is a secondary absence of integrin in this case. We know that the integrin is also absent. So these two complexes go together, and when you're missing one, the other one is also missing. Dystrophin is still present, so there is some mechanism.
Turn your video off so you're not
okay. Surrounded around. But interesting, the integrin is missing. And integrin, we know, is really critical for cell communication, especially cell survival. So without the linkage to integrin, you're actually missing the AKT, which is a signaling pathway activation, and therefore you're missing that muscle cell survival. So when the matrix is present, muscle cells like to know that they've got a friendly matrix around it and sending signals into the cell that says, stay alive, grow, survive, everything is good. But when that integrin complex is missing, you miss those signals into the cell, and therefore the cell tends to not grow and tends to be much, much smaller than what it should be. I talked about laminin 111, which is the 111, and this is the target that we're using right now, although our cell lines could produce 211 as well if we needed to, and I'll talk about that in a second. So in this case, actually alpha-7 beta-1 loves laminin 111. It's like its favorite matrix protein. It likes laminin 211, but actually likes 111 even better. And so when you put 111 into the system, it restores the laminin back, the integrin back onto the membrane, and you start these cell signaling pathways that promote cell growth and survival. So why use 111 versus 211? So patients are missing 211 from birth, and if you put 211 back in, what do you think is the major problem that the patient could face? an immune response because they've never seen this protein before. And so if we use 211, the problem is the immune response could be triggered to target, to target the 211 and clear it out. So it's called, it's basically an immune deficiency of the laminin. So the great thing about 111, lamina 111, although it's missing in the muscle, it's present in our kidneys and many other tissues. And so it's not recognized as foreign, right? So the key thing here is, can 111 really replace everything that 211 does? Why does our muscle in a normal situation or unaffected person like 211, and why is that important? And I might have some tricks, some ideas about why that is happening in a couple of slides. So that's the reason why we've chosen 111 over 211 at this point, although our cell lines could produce both, either or. So we could go either way. And especially if we, so if we use 211, we'll probably have to use some kind of immune suppression. 111, we don't need to do that. So these are the first kind of experiments. They were done by a graduate student at the time, Jacinta Rooney, who's the lead author on this paper. She actually works with Carsten in Carsten's lab now, and so she's working there. And these were some experiments where we were taking mouse laminin, and so this is the mouse laminin, which is here, and she injected it. And what we were really interested at the time was what with the laminin, which is a big protein, would it just sort of like form in an area, and then you would get maybe some repair, because we're interested in muscle repair at that stage. And she came to the lab meeting and said, it's gone throughout the muscle. And I said, that's insane. Since 900 kilodalton protein, it's huge. That doesn't make sense. And so I told her to go back and repeat it. And she said, the next lab meeting, it had done the same thing. And so this is the result she had. So this is wild type muscle, which has laminin alpha 2, the adult form. And it's missing in the mouse model that we're using here, the DYW mouse. And of course, we don't have any lamina 111 present in the muscle because it's missing in the adult. And then this is looking for lamina alpha 1, and you can see that when she injected it, it went all the way around the muscle fibers. So it looks, a lot like this in terms of the distribution. And we use this as a control spectrum just to make sure that the muscle is good for immunofluorescence. And then she was able to follow the laminin from the tube. So this is the laminin in the tube, and it goes through the serum, and this is systemic delivery, and ends up in the muscle tissue, which was really surprising to us. I mean, I didn't expect that to happen at all. So that was kind of the first set of experiments. So they give you some history. And then, of course, we did a lot of experience looking at survival. So this is actually the lamina alpha 2 knockout mouse. This is injected with just vehicle. This is injected with the laminin protein. So these are the same age. And you can see automatically there's a huge difference in how the mice look. And we have a video with this paper. So if you want to look at it. And then these mice, the lamina alpha 2 mice, this is their survival curves, die at very young age. When you treat with lamina 111, you can extend this out, and this is wild type at the top here. So we had a highly significant increase in survival in these animals. And this is a lamina alpha 2 mouse. Now, mice normally live about 2 years. So now we're good at 60 weeks of age. So we're over a year. So we've really substantially increased the time frame, the survival. So more recent studies we've done is to look at laminin after disease onset. So most patients are diagnosed, and this, yeah.
I have one question. We said, can you explain what you mean by wild type?
So wild type is like the unaffected. Okay.
And I had a question too. At what age did you apply?
Did we treat?
Yeah, what age did
you so these were treated all at about 10 days of age for a mouse. Okay, 10 to 12 days. So pretty young. So the big question is now, what if we treat later, right? Because most patients, most kids, have had the disease for a little while. So what happens? And this paper describes, this is actually done by Pamela, who was a graduate student in my lab and is now in Boston, the Boston Children's Hospital, in which she did injections of mouse laminin after the disease had already progressed. And what we found in that instance is that the laminin got to the muscle, just like we saw before, and it held the muscle at the current level. So when we look at what was happening, we were hoping it would reverse things and actually promote muscle repair and regenerate muscle. It stabilized the muscle. So it didn't get any worse, but it kind of kept it at that level, right? So the key thing behind this paper, which you published in 2020, was that the muscle got stabilized as in the current form and it didn't get any worse. So that's the main thing. We're hoping it would get better, but I can talk about what the role that 111 may be playing in muscle repair in a second. So now this is human recombinant laminin. So we've got a cell line, it's transfected in with 3 constructs, each of the human chains, alpha 1, beta 1, gamma 1. And then, so we actually can do a Western blot and show that we have alpha 1 and beta 1, gamma 1, which are these chains are right close together. And this is a picture of our molecule. So it forms the typical cruciform shape right here. So this is actually it here. These are all the other molecules that are in there. And you can see it produces a nice cruciform shape. So we know we're producing the heterotrimeric protein from these cells transfected in, and we're producing a lot of it. So we have enough now, and actually for Haley in my lab, that's really great because she can actually have so much laminin now she can treat it. And the great thing is the laminin is really, we've got a purity thing. So the first major problem in this project was production and purification, and those were solved by Prothelia. So we got a really good cell line, produces a lot of laminin protein, in fact, much more than we can use now, and Haley has gobs of it in the freezer, and it's highly pure. So the purity is between 98% up to about 95% in each of the batches. And so this actually shows the purity, and it's mainly this guy here, this laminin heterotrimer, okay? A few little fragments around, but mostly pure. So when we did this rotary image shadow imaging, Prothelia was super excited because you can actually see your therapeutic, which is kind of cool. So Haley has taken that protein and she's actually labeled it, and we can actually follow it as it goes into the mouse. So this is the mouse model for laminin alpha 2, and this is him sitting on his back and then on his tummy. So we can actually see where the laminin is going after we inject. And this is the time after injection. So 30 minutes after injection, 1 hour. And you can see pretty rapidly, even 30 minutes, we're starting to see muscle localization of the laminin. We have this really hot spot here, which turns out to be the liver. So we know that clearance is occurring in the liver. And when Haley actually took the organs out and she could image them. So we know that a lot of protein gets trafficked to the liver, which is probably then getting degraded. But we get fairly rapid accumulation. And over time, 4 hours, you can see the muscles are really beginning to light up. And this is the bladder. So we also lose it through the kidneys. So this is kind of like part of drug development. We want to know where it's going and how it's being eliminated, and maybe how we can actually improve the retention of the laminin within the muscle. And this is where he's sitting on his tummy, and we're looking on his back, and you can see the muscles lighting up. And then you can actually see some of the kidneys beginning to light up here. So there is loss of the laminin, and this is 10 milligrams per kilogram. Interestingly, a mouse rapidly has a system that more rapidly clears drugs than humans. So that's one thing we have to know, that in humans it's probably going to be a slower process. Yeah. So the laminin, Haley labeled the laminin with a fluorochrome, and then was injected IV.
The question was, how was it injected? Just for everyone.
Yeah, so injected IV. So it actually went. She injects. There's a vein in the back of the eye, and she injects right behind the eye and into the vein. So these guys were injected. How old were they injected, Haley? Yeah, 4 weeks of age. Okay, and so then she actually took the muscle from this mouse and then looked a week later. So this is the diaphragm muscle, and you can see the human laminin in the diaphragm, the TA and the gastrocnemius muscle, and the vehicle treated didn't have anything. Okay, so that doesn't have anything. So it tracked pretty well and localized to the muscle. So this is the drug that will be used in patients once we get it to the appropriate purity, right? And that's an important thing. So as I mentioned about satellite cell repair, so what happens in patients without laminin, in our normal muscles, we have these stem cells, and they can repair muscle. And in fact, in some dystrophies like Duchenne muscular dystrophy, those repair cells actually can repair for a while, and then they kind of exhaust themselves. But in laminin alpha 2, they don't have this matrix. The matrix is really critical, and it kind of supports the repair of muscle. They don't have that, and so therefore they have a failure of muscle repair, which also contributes to the disease. So we wanted to look and see this environment. So what's happening? So we actually could isolate these myofibers. We can actually tease them apart, and then we can stain them for, and so we actually treated the mouse with either recombinant human laminin or 111 or recombinant human laminin 211. So now we're looking at the direct replacement versus what. So we wanted to try and tease out what could be different between the two laminins, and why are we going to miss something when we use 111 versus 211? So this actually shows the satellite cells, which are these guys here. We've marked them with this with PAC7, so you can actually see the satellite cells in both mice that have been treated. And then we looked at the laminin and its distribution around the satellite cell. So one big difference you notice is that 111 tends to cover, yeah. Okay, satellite cell is a repair cell. So it's a stem cell that helps repair muscle. Okay, all of our muscles. So when you go to work out, you damage your muscles a little bit when you're working out, you can actually repair your muscles with these stem cells. Okay. So interestingly, when we inject 111, what we get is the satellite cell niche, that area that the satellite cell is, gets covered by 111. And so you can kind of see the overlay here. Where if you use 211, 211 tends to go into underneath between the muscle cell and the satellite cell. So it doesn't cover it, it tends to go into that little niche, which is critical for actually repair, because that region where the satellite cell or the stem cell is, it needs to talk to the muscle cell through the matrix. And so the worry that we have here is that although we're getting 111 coverage, we might be missing its ability to repair. In fact, what we've shown is that satellite cells or stem cells, when they repair muscle, they actually undergo a symmetric or asymmetric division. Asymmetric division means that they divide and they repair the muscle, and the other satellite cell replaces the one that's just been used. So that's good, and that's what 211 does. In the case of 111, what we see is it just goes, both cells go to repair the muscle, and we don't replace our satellite cells or our stem cells. And so eventually we would exhaust with 111 our stem cells. So that's the difference that we've noticed in our counts. So 111 seems to promote this repair and not replenishment, 211 repair and replenishment. So that's the difference we've found so far. So you know, there's the good and bad, right? There's the compromise that you've got to have sometimes. Yeah.
So that's basically why you're seeing stabilization and not regeneration.
You get stabilization. We do get regeneration, but we eventually exhaust that, is what we think could happen over time. Exactly. So yeah, we were hoping we would be able to completely regenerate the muscle again, but it looks like it kind of exhausts that possibility. So that's probably why we have 211 in our muscle, at least one part of it, and not 111. That's why it kind of turns off. And the other thing could be a structural component, that 211 tends to be a lot stronger in its interactions with the matrix, and 111 may be a little bit more labile. It might be a little bit more for mobility of cells more than necessarily the structural component. And that's why it's a stronger structural protein. It's like reinforcing your concrete, right? It's a reinforcing component. Okay. So the other thing that Haley has done now is we've used a new method called digital spatial profiling. This is a brand new technology that Haley has been using to actually look at simultaneously, how does laminin 111 in this case restore all of the parts of muscle? So with this technology, and she used not the transcriptome, but she used the antibodies, we can screen literally almost 100 antibodies simultaneously within a couple of days across the entire muscle. And so what she did here was she took the muscle, and actually where you see brown here is the laminin. It's being labeled with an antibody, and so we can actually see the brown staining around the muscle fibers. So again, the laminin is in the muscle, in the injected mouse. And then we use these morphological markers, Desmond, DNA, and these markers here to actually identify muscle versus like inflammatory cells and so forth. And then what we could do is then look at particular regions in the muscle to look at either inflammation regions or regions of what appears to be just normal, not normal muscle, but muscle that is intact, more intact muscle, and look at the expression of a whole range of different proteins at once, including signaling pathways like the survival signaling pathways that integrin activates on, immunological markers, and we can do this within one slide simultaneously. This technology is used in hospitals now to actually type cancer cells. So if you go in to want to have some kind of chemotherapy for a cancer, they actually do this to type the cells so that they know what type of chemotherapy to give you. We've adapted it now for doing in muscle, and we would like to get it. Actually, we're applying to NIH to try and get a muscle-specific panel because these markers are for signaling pathways in survival, inflammation, and fibrosis mainly, which is the main components of the disease, but there are other muscle-specific markers we look at. And so when she looked at this, the good thing about this is we can identify also biomarkers, which is the next critical thing that we need to do, especially going to critical clinical trials. These are just a few of them, but we saw that laminin 111 restored in the muscle using this technology, many of the markers that are associated with metabolic changes that occur in muscle, so it restored it back to normal. Markers involved in cell survival. Again, we would expect that. The AKT signaling pathways I mentioned should be restored. And so we get a lot of restoration of those markers pretty well. But some interesting markers that we didn't expect that change. So this is the normal unaffected mouse. This is the diseased mouse without any treatment. And this is the mouse with treatment. And you can see laminin restored it pretty much back. And this is called P-junk, which is part of a pathway in muscle, restored it completely back to normal with this. And this is only with 48 hours treatment. So very rapidly, we restore the muscle back to a sort of homeostasis, its normal, much better state. We do get an increase in fibronectin in this case, but this is within 48 hours of treatment as well with the laminin treatment. So this paper is actually Haley's currently writing it, and so we're hoping to be able to get this information out. But it's a really rapid way to screen through and actually look at the effects of it. And we want to do this also with recombinant human laminin 1, laminin 2, and see what the differences are. So the great thing about the cell line, as I mentioned, we can produce 111, which is what we're going for now, or we could transfect in the laminin alpha 2 and produce 211 and actually do a direct replacement therapy. So in terms of like where we are now, I kind of want to talk about where we're going right now. We're optimizing the vehicle component for delivery. That's the big thing that's happening right now. And so that's the next sort of big phase. And then we want to redo that, sort of do experiments in sort of larger animals, to be able to scale this up so that we get towards patients. And then we also need to identify biomarkers. So it's really critical when we go into the clinic, and Anne probably can attest to this, is that, and probably Carsten, and Carsten actually talked about this, if we don't have a biomarker that we can test to see how effective our therapy is, then we're kind of in the dark, right? So we want to be able to say, okay, we're either looking at creatine kinase, or we're looking at some marker or a muscle marker that says, okay, things have been restored or improved greatly, and then we can measure the efficacy of our therapy. So yeah.
2 questions. The first one was, how long can the satellite cells repair before they are depleted in the laminin 111 case? So
we don't know the answer. I don't know the answer to that yet. We're carrying on experiments with it, but it would suggest that yes, once the laminin is present, even if it's 111, it will begin the repair process, but it will exhaust over a period of time. We haven't done the direct comparison where, okay, if we look out over a longitudinal period of time, how long it takes to exhaust. So we don't know the answer to that question. That's a great question that needs to be answered. Again, one of the things I would like to emphasize is that it's great to have your technology taken by a company and developed, but we still need the basic science to move along with it so we can understand exactly how the therapy is being developed.
Okay. The second question is, can you elaborate on what you're doing to optimize the vehicle for delivery? What contributes to optimal versus non-optimal?
Yeah, so I mean, it really depends. So in the mouse studies, with this current vehicle, this current solution it's dissolved in, if you like, we saw no negative outcomes, right? So literally thousands of mice have been injected. They've been repeated by independent groups. So these studies have been independently verified, showing improvement of muscle force. So all of that has happened already, right? So we know this works, but we want to optimize it for human human delivery, right? For human patients. So we want to make sure that that it's going to be safe, and that's really the critical thing. Is I think, as Carsten would say, we want to know that we want any therapy developed to be absolutely safe for the patient. It doesn't cause anything to be worse, right? That would be the worst thing. So the important thing is to optimize everything for patients.
Cool. We've got 2 more questions.
Okay.
Would laminin 111 improve peripheral neuropathy or just muscle?
Perfect. That's a perfect question. We don't know the answer to that. I have a grant in with NIH to absolutely answer that question. And Haley has been beginning to inject into the cerebral spinal space because laminin doesn't cross the blood-brain barrier. It's too big a molecule. So she's begun experiments where she's injecting into the cerebral spinal space. So she starts in the sort of lumbar region injection, and she's got beautiful images, just like you saw with the mouse here. Oops. Showing the human laminin distributing up and down the spine and then out to the peripheral nerves. So we think that we hope that will be a better delivery method, since we can't get it into that space. It's kind of an immune privileged space, right? Although I think in mouse and patients, there is some leakiness in the blood-brain barrier, but laminin does not cross that. It's too big.
I'm thinking we need subtitles for the Australian and the New Zealander talking.
There we go. Yeah, let's go.
Hopefully everyone can understand us. The next question is a combination of both laminins a possibility? I think they mean laminin 1-on-1 and 2-on-1.
And 2-on-1, it's definitely a possibility. Yeah, I mean, it's always difficult when you're mixing drugs, and I think that Carsten and Anne probably could talk this. You have to optimize both and meet the FDA standards for injection. So it's always hard when you get multiple drug treatments, because you know, are we going to use, you know, how many milligrams of this per kilogram versus the other one, and how do we mix that together? So it's always difficult. Or do we use 1-1-1 and then switch to 2-on-1, like which naturally happens in your muscle during development? And that might be a cool strategy, actually. So, you know, where you start with 1-1-1, get things going, and then switch to 2-on-1, so you preserve those satellite cells. Very great point, but we don't know the answer to it. Sorry, something I would love to get funded to be able to do. So
maybe I have a question for you, Dean. So a lot of it is in the liver. So that's
yes,
is a great tissue to re-extract it from and see what its integrity is. Do you get the full heterotrimer out of liver or of any tissue?
I don't know. And that's something I think that Haley would want to look at, I think, to extract it out and look at, see if we still got it, or it's beginning to be degraded. And actually that's a method of, there is some in the heart too. So that we know we're targeting the heart tissue. So when she pulls the heart out, but the liver is the hottest organ there in terms of where the drug is. So you're right. Is it like a sink and it's sitting there and like being released over time, or is it actually being actively degraded through the liver as you get?
Yeah. Also, the interesting question is whether the entire full big heterotrimer actually gets to muscle in its full integrity.
Yeah.
Do we know
exactly.
That,
so it looks like from our Western studies, it looks like full length is there, but there are definitely some fragments. So you do get a breakdown. I think what happens is, so in the mouse model and probably in patients too, there's a lot of proteases in that matrix that break and remodel the matrix. And so what we've been able to show is that the laminas, you first go in, so they're like, you know, you get the first dose, it starts to stabilize those proteases that are breaking down the matrix. And then with more doses, it gets more and more stable. So it takes a little bit of time to like stop the active degeneration of the matrix and the remodeling before.
Yeah, that gets me to my other question, which is a human translation question.
Yeah.
So in the human patient, we have, the mouse is a great model, but in the human patient, there's even more fibrosis and connective tissue in the muscle that you have to kind of get through.
Penetrate into,
to get to.
No, I agree. I mean, you know, we've been able to solve a lot of mouse muscular dystrophies over the years. Just look at the Duchenne story, right? There's a lot of mice that can be solved, but things didn't translate that well to humans. And so that's one of the things that we've got some of the key research scientists here who are working on other approaches as well. Whether all of this, you know, whether this one goes or the other ones, I think we just want to take the best thing forward as we move forward. Nothing's going to be simultaneously, unfortunately, developed, so you can pick and choose and say, okay, I'm going to go for a gene therapy approach or a linker approach or a protein therapy approach. That's going to be the thing, because we're not all at the same stage of development. And the question would be, okay, when we get to this point, then I think people like Carsten and Anne then have to kind of decide, okay, which looks like the most efficacious and the most safe to move forward. And I think safety is our primary goal here. We don't want to make things worse, right? That's the critical thing. So we want to make sure what we're doing is a safe approach that also shows efficacy as well. Yeah. Well, thank you.
Have you checked whether 2-1-1 can also restore?
So no, that's a great question. That's the next experiment we would I'd like Haley to do. So yeah, I've got again, the one thing you can really help was like contact NIH and say support funding for myristoyl deficient congenital muscular dystrophy. I mean, that's the big thing, because basic research, you know, we get some funding from the companies, but their money is going all towards, you know, production, safety testing and that sort of thing. They don't really have a lot of money to invest in basic research, but we need that at the same time. So, you know, contact NIH and let them know that you need to have the work funded, because that's how myself and Dee, we get funded and able to do the basic research to bring the, you know, to make sure that we understand the biology, because good science leads to good therapies, you know, so that's important.
We have one probably last question. I think we have to then move on to the next presentation, but this is probably way too early, but if this will work, how many times do you expect you have to inject laminin into the patient?
So at the moment, it looks like on the initial testing, the area under the curve, which tells you about the dosing, is really good. So we can actually probably reduce it down. So these mice were getting 10 milligrams per kilogram per week. So but they metabolize the drug much faster than people. So we're hoping that we can get it down to, we don't know exactly what the dose is because that needs to be tested, but we're aiming for once a month would be an appropriate infusion time. But the longer the better. And we know that we can build laminin. It's almost like you could have a loading dose and then a maintenance dose. And the maintenance dose loading dose to stop everything and hold it where it is, and then a maintenance dose, which could be once a month or once every few months. It's possible, yeah. So then maybe you might be able to get it injectable at home. So actually, Haley's done lots of different ways of routes of administration, and she did sub-Q. So that was one of the things that she's done. So there are some routes that are not that great, and there's some for delivery, and there's some routes that are really, really good, right? So yeah, we're hoping we maintain, once we get the maintenance dose, it's going to be less frequent, and then we're just going to maintain. The matrix, once it's there and stabilized, it's pretty stable for a period of time before it goes over. But we don't know exactly the time point of that for now.
Cool. Thank you, Dean.
You're welcome.
My name is Judith Kemaladewi. I am an assistant professor at the University of Pittsburgh. So I got to this field in 2015, I guess, when I started doing my postdoc in Dr. Ronnie Cohn's lab in Toronto. And actually, no, okay, let me backtrack. So I started my postdoc in 2012, and around the same time, CRISPR became hot. I originally was supposed to do something else, but then because CRISPR became hot and my postdoc mentor just told everybody, let's work on this thing. So in the beginning, we used CRISPR-Cas9 to correct mutation in a mouse. So one of the mouse models called DY2J have a mutation that affects the splicing and leads to disruption of the normal LAMA2. So we learned all the nitty-gritty about the CRISPR-Cas9 technology, how to deliver it to the mouse model, whether it's needed for both muscle and nerve, or muscle only or nerve only, and whatnot. And we got it working very well. But then also, I think when I started to come to the patient organization conferences, I sort of like pitched to everybody in the room if people are willing to donate some cells to our lab. And I think the Bert family was one of the first families who jumped to the thank you very much. And we started to realize that the mutations that affect patient populations are very heterogeneous. So if you want to correct each of the mutation one by one, that is not impossible, but it's difficult. It takes a long time and takes a lot of efforts to actually do that. So around the same time, there's also the 2.0 version of CRISPR technology that doesn't cut, but then you can still use some of the properties to bring something to a part of DNA. So we use that to kind of like wake up laminin alpha 1, which is part of the laminin 111 that you just heard about, that is supposedly silenced during silenced during postnatal stage. So it's only turned on during embryonic. So we use CRISPR activation, we call it like that, to kind of like wake up the sleeping giants, if you will. We used kind of like a similar delivery technique as how we deliver the original CRISPR-Cas9, which is using AAV9, adeno-associated viral vector 9. And because the
can you explain that? That's a virus, right? You use like a benign virus to transfer through the body instead of an injection. Is that right?
So it's a benign virus that we can inject into the body, and it has a certain packaging capacity. This is also what is used in microdystrophin trial because it targets skeletal muscles and neurons quite well, I believe. Yeah, it's one of those things that are accepted in the neuromuscular field. I must say, we just pick the one that people use. Yeah, so we package all the CRISPR activation component to wake up LAMA1, to wake up LAMA1, and we tried them in a mouse model for LAMA2 deficiency. And we were able to wake up this LAMA1 in both muscles and nerves, and that went very well. But the problem with this kind of like technology is that the AAV9 has only limited packaging capacity. So we had to use 2 viruses to carry all the different CRISPR-Cas9 components. So moving forward, our goal is to bring down the 2 AAVs to 1 AAV so that we can reduce the cost as well as the potential toxicity. And that is what my lab is busy working on right now. Thank you, Carsten, in advance. For whatever you are going to say.
Analogy for this type of approach is if you think about a word processor, and you have a big Word document, you think about a word processor and you want to go somewhere in your huge document, you do the find function, right? You open that find window, you type in where you want to go, and the word processor finds exactly in the document where you want to go. So that's what CRISPR does. But now instead of deleting the word that you find, you bold it, right? So this is the CRISPR upregulation. So it's basically in simple terms, it's a search function, you go exactly where you want to be, and then you click on bold, make it big. And that's kind of is a laminin 111 approach, the laminin alpha-1 upregulation approach, in simple terms.
Are you able to use that same analogy to try to explain gene therapy?
So gene therapy is a very broad term. What it encompasses many things that are directed at genes for therapy. But what it currently means is using AAV, adeno-associated virus. Let me just give you a few pointers to adeno-associated viruses because it comes up in a number of these approaches. So adeno-associated virus is a very small virus that is not causing disease in humans, really. It infects humans, but it doesn't cause disease. It can't even act on its own. It has to be carried along by a bigger virus, which is adenovirus. That's why it's called adeno-associated virus, because it's incomplete. It needs helpers to do an infection. But because it is good at infecting and not pathogenic per se, it doesn't cause a disease per se, it offered itself basically to the gene therapy field as an envelope to deliver something that's not the virus's own genome. Every virus has a genome encapsulated in an envelope that's made of proteins. And a virus normally uses the envelope to deliver the gene that it has to the cell and coax the cell in making more of the virus. So that's why we get a cold and an infection is because a virus goes, delivers the genes take over and make more virus. So the trick here is to take the envelope to deliver something, but it's not the viral genes, it's actually a gene that we want to get into the cell. So we cripple the virus in delivering, but not replicating in the tissue. That's the idea. And so the technology that's now being developed is to have production cells that make all the viral components that are necessary to make an envelope, but insert a gene of interest that is then delivered. And that gene of interest can be anything. It can be a gene replacement, it can be a CRISPR component, it can be a gene regulatory part. The drawback is the capacity of this envelope is small. It's a small envelope. It's not a big kind of FedEx envelope. It's a small envelope. So you can only get, yeah. So you have to really think about what you write on your card and want to send there, because you can't deliver a novel. It has to be a poem, if you will. Yeah.
It should be a tweet instead of a.
And so that's a challenge for the gene therapy field at the moment. So the and that's why Dewey had to use 2 viruses initially, and Dewey is working on narrowing that down into deliverable in 1 virus, as is Modalis, which is using a similar approach. The current envelopes, there's a whole different colors of envelopes or different versions of envelopes. Most of them currently used are natural envelopes, so that occur in nature, that people have isolated from humans or animals and characterize what they infect. There's a natural immunity, of course, to some of these. So some people have been infected by AAV without even knowing. And so probably 30% of folks in the room have antibodies against a given serotype that doesn't allow that to be used as a treatment. So they're already immune against it as a treatment. So that's one problem. And the other problem is that one had to use very, very high doses of virus to really get to all muscles that are needed to be transduced, and that causes its own set of problems, including immunological problems. And so there's a new avant-garde of making envelopes that are now tailor-made, where the address is written more precisely so that they go to muscle more specifically, so that you can reduce the total dose and de-risk the gene therapy by making it more directed to the tissue that you want to go to. And so that's been presented at this meeting as well.
Yeah.
So it's a tool that one can use quite readily for different applications with the constraints I mentioned.
Carsten, there's a question kind of related to that. I think it's a little bit different what you just answered, and it's asking if you were to use AAV as a delivery mechanism for a drug that needs to be delivered every month, is there a chance you could become immune to the AAV?
Unless you do tricks of trying to trick the immune system to forget about it, or use a different envelope, but that's currently a really relevant problem. You cannot redose at the current time after you. So your first shot on goal has to be really good.
So other thing that we package into this AAV is the Cas9 or dCas9, as you will, and that is also another gene or protein that we might have antibody against because that came from bacterial infection.
Yeah, that's
so,
a good point.
Yeah. So that is another layer of complication that one needs to address moving forward.
Yeah, so that brings up a really good point. So there's two immunological layers to gene therapy. One is the immunity or the reaction against the envelope. That if you have neutralizing antibodies against the envelope, you can't even get it into the cells to start with. This patient is already immune. If the patient has been treated before, you can't redose because of antibodies. If the dose is very high, the immune system can get upset and cause adverse problems. But the second problem is the immunological consequences of what you deliver with the AAV. So in the case of CRISPR-Cas9 proteins, these are proteins that the body doesn't make normally. They don't belong to the body. And so if you inject it with the AAV and it's being made by the cells, it's a foreign protein. And that could cause immunological problems in its own right. Yeah. Similarly, if you deliver a gene that's missing in the body because of disease, so someone doesn't have it, like laminin alpha 2 in this case, if you were to deliver new laminin alpha 2, the body would think, oh, what is this? I've never seen this, and may reject it, just like in an organ transplantation where you reject an organ. It's just a single molecule that's transplanted here. And so that's why Dean is using laminin 111. That's why upregulation of laminin alpha 1 is immunologically favorable, because that's the body's gene. The linker proteins that Judith we're talking about, they are basically engineered versions of the body's own protein. So that is probably also immunologically favorable. Yeah.
I have a couple of questions here from the meeting chat. The first one is, how long does the CRISPR remain associated with the DNA? I'm not sure.
Remain associated with the DNA. Can I answer?
Because CRISPR is effectively, like Dr. Bonnemann was saying, it's just the ability to find the correct area to make a change or upregulate or whatever. But does it stay around DNA after it has done its work to find the correct area, or is it dissipate after that?
Let me think about that.
We may be misunderstanding exactly what CRISPR does. I'm not sure.
No, that's a fair point, but I have to go back to really the biochemistry of the CRISPR-Cas9 that I am not very much expert on. I can answer it in another way probably. So the Cas9 can linger in the mouse body for a while, so it doesn't really get degraded for some reasons. So the longest that I've seen, at least the Cas9, because there are multiple Cas9s as well from like different bacteria. that I've seen are probably about 1.5 years, and that is from GM1 myotonic dystrophy mouse model. Now, there might also be, because the muscles degenerate at different pace across different diseases. So that might also affect how long it lingers in each disease model.
This is actually an interesting question because, of course, the CRISPR search function gets you there, and then you can do different things. So initially, CRISPR was the gene scissor only. You just cut the gene. So there you can say, well, I only need CRISPR for the cut, and then it can go away, and then we don't need it. Actually, we don't want it anymore. In your approach and Modalis approach, you will have it want to stick around, right?
Yeah,
because you want to continue. Yeah. So that's an important.
Yeah. So people want to shut off the ability of Cas9 to cut, but we kind of want it to linger a little bit longer.
Because you're not cutting.
Because we're not cutting, and we kind of want it to be sustainable in upregulating LAMA1.
Exactly. It needs to be sitting there on the regulator of LAMA1, keep cranking it up.
I'll ask the more recent question first because it relates to what was just asked. Is the idea that a one dose does the job permanently in these types of, I guess, in the AAV delivery? The goal is, you're saying you want to have a great shot on goal because you get one chance. Is that the idea here?
Yeah. So yes, that is the idea, but it is kind of limited by biology because whatever the virus delivers to the cells is not integrated into the cell's own chromosomes. So it is not, it doesn't become part of the genetic material of the cell that is inherited. So when the cell divides in the chromosome, this is passed on to subsequent generations of the cell. However, what is delivered with AAV lives in the cell nucleus, but as its own genetic material that is not inherited. So what that means is, if muscle turns over, if muscle cells duplicate and replicate, if muscle growth, if muscle regenerates, there's a chance that your effect gets diluted out, gets lost by growth and regeneration of the muscle. That's good because you want it to regenerate and grow, but it's bad because you're losing your therapeutic effect along the line. As opposed to a cell that doesn't divide anymore, for instance, like a nerve cell. There you don't have to worry about it because then it's good as long as a nerve cell is alive and it doesn't need. So all the more
I think the gene therapy field in general is also addressing that, whether you're doing it for other things as well. Yeah.
Another related question is, can anyone explain, because there was the SMA drug that was recently approved by the FDA. I think it was the famous $1 million dose drug. Can you explain how that works and how that may or may not be similar to what we've been discussing here?
It's similar and different. So in SMA, you're really missing the working gene product. And the working gene product is small enough to fit into AAV. Not only that, there is a backup copy of this missing gene product that's just not perfect. It doesn't really work, but it's immunologically similar. So now you can put in the perfect version of the gene. The body already has knowledge of it, although imperfectly, and you put it into neurons, into nerve cells. So these are cells that no longer divide. So once you get in there, it really has a great chance of being durable and correcting that neuron, if it's still alive, for, I don't know, decades, but for a long period of time. So it's different in that these cells are no longer turning over. They have terminal cells, they're final cells, and you have a gene product that's missing, that's small enough to fit into AAV. So it's kind of the perfect situation for gene therapy. It has, of course, been a resounding success, but can't be the standard for every gene therapy coming after it. It's just other diseases are different.
2 questions from the room.
So when our son was born, we had a diagnosis pretty quick, within the first 2 months of life, and he was at Hopkins, and Dr. Crawford actually, I guess, used to work with Dr. Cohen. So we heard a lot about the CRISPR early on and so on and so forth. And they really were optimistic because simultaneously Spinraza, they were on the team with that. And their best guess was like within 5 years there would be some type of cure treatment. And I guess what I'm wondering, Micah turns 5 in 2 weeks. And I'm wondering if there's like where you would stand now as far as hope and like the timeline, and are we any closer than we were, 5 years ago?
Fair question.
Fair question, yes.
The answer is complicated but hopeful. I think we have this meeting. I mean, there's almost an embarrassment of riches of therapeutic approaches to LAMA2 that we've never seen before. I mean, this is just extraordinary. So just this room alone offers like 5 different approaches to treating this ultra rare severe disease. I think that's remarkable, really remarkable. And the other thing is that the development that leads us closer to treatment is not for LAMA2 alone. It's in the entire field of muscle disease. So that's another thing that I tried to point out earlier, is we're not doing our own thing alone and trying to reinvent everything that's needed. The entire toolbox that we need to treat these children is growing with us, and we can take advantage of it and learn from other applications. We learn how to give an AAV gene therapy. We learn how to treat the immune system. We learn how to package and manufacture properly. So that is kind of exponentially advancing our approach to the clinic. However, they're just big unknowns. Any therapy can run into sudden problems that are not anticipated, that were not predicted, that are human problems alone. And so you can not know what type of roadblock you might hit and what type of diversion you have to take to get to the goal. But I think, and that's why it's so hard to put years on your on your answer. But I can assure you that we've never been closer, and so I think I'm an optimist. No, I'm pessimist, but here I'm I'm optimist. So
one thing that I can assure you, from the perspective of early career investigator, is that I am committed to do this.
So I just wanted to try to understand everything that's going on here. So the laminin 111 that he was talking about, it seems like it's something that's produced in the body, but it's produced elsewhere. So it's something that the body is familiar with, and you're using it to essentially inject it using an IV. And then your philosophy, you're using CRISPR-9 to kind of wake the laminin 111 that's already in our body.
Yeah.
And it seems like the two different approaches are, we would be getting 1 month-ish injections, and this one, maybe would last longer, but you would still probably have to be redosed, which the complications of that is that you can't use the same virus. You might have to use a different one, or might have to trick the body to re-entering it. And then the question I guess I had is, you were talking about laminin 211, which I guess is not in the body. I'm not sure like where you found that or how you produce that and where that's coming from. And since it's not in the body, like the CRISPR thing isn't going to work for that, because it's not something that you can turn on like the giant and the LAMA 111, because it's not really there to begin with. Is that right?
Correct. Yeah.
So in the patients, they have mutations in the gene. So turning on or upregulating laminin alpha 2 is not going to help the situation, right? Unless you correct the mutation, you've got a non-functional laminin alpha 2 gene, right? So no matter what, that's going to be problematic. So that's why and of course it's present throughout the muscle. It's all, every gene is mutated. So that's why they don't produce that. So that's why we've gone with 211. In our case, we could use a replacement therapy, but as Carsten mentioned, the worry would be the immune response to what would be the foreign protein in the patient now. Well, I mean, it wouldn't help unless you edited it using CRISPR.
So my question, as we learned yesterday, that LAMA2 with all the patients kind of present differently. I have 2 sons with laminin 2, and they present differently. And I know early on when they were little, their neurologist she always wanted to take pictures. Like it boggled her mind that they presented so differently. So now my question is, when it comes to when these therapies maybe start to work and you start giving them to patients, does the differences in the way that they present or their severity, will that affect the effectiveness of it?
It's a really great question. So there's two things to consider in this. So even patients in the same family, I don't know about your children, with the same genetic mutation may present differently. And that is because they're different genetically, and the mutation plays out on a different background, and there's different genetic backgrounds that make a patient more vulnerable or more resistant to the primary genetic defect. And that's something we are in the field are trying to really figure out, because there could be clues in how to ameliorate the disease apart from that. But it does not invalidate that the same treatment would apply for both of them. So the treatment would still apply just the same way. And so we don't have to kind of subdivide our patients with great granularity. We want treatments to be approachable for all patients, no matter what the severity. May I make a suggestion, allow other scientists to introduce themselves as well that are in the room? Judith, you want to go next? And who else would like to speak? Okay.
Yeah, so I quickly introduce myself. So my name is Judith Reinhardt, and actually I decided to show you slides now, because for me also English is not my native language, so it's actually easier to actually for me to present what I mean when I show you some illustrations for this. And so I work in Switzerland, and I work in the research group of Markus Rüegg, and this research group is working with mice that have LAMA2 deficiency since more than 20 years. And also has contributed with these mice a lot about the understanding of what is actually going on when laminin are missing. And I think you all know these illustrations very well, and we all draw it a little bit differently because we want to point out different important functions of these laminin proteins. And here I illustrate that these laminins, so down here would be the muscle fiber, and on top you have this shield of the muscle fiber, which we call extracellular matrix, and it protects the muscle fiber from being damaged when the muscle fiber contracts. And this laminin sheet is very essential, and it actually becomes apparent how essential it is by laminin deficiency. And it's essential that this laminin protein is linked to the fiber, to the muscle fiber, and it does this by these two proteins here. We heard from Dean about integrin, and this is another protein here, dystroglycan, that helps to anchor this laminin to the muscle fiber. The other important point is that these laminins actually bind to each other, and only when they very stably bind to each other, then it can form a scaffold which is very, very stable. And if they don't do this, then they form some kind of labile scaffold, which is not that good as the one which you usually found with laminin 211. And this is then more the drawing you are familiar with, and this also illustrates one of actually the big challenges we all facing with this disease. So you all know that the mutations are in the LAMA2 gene, but this LAMA2 gene would produce a protein that is just this guy here. But what we need in the end is this cross here, and to form this cross, you need additional proteins that the body produces, and actually the body also of LAMA2 patients produces these additional proteins. But because this guy is missing, all this thing here falls apart. But this guy here, we know from the mice, and also we know from the human patients for what it is important for, but this guy has huge challenges in terms of to bring it into the muscle. And I think we've learned now from Dean and from Dwi and also from the other researchers, we all try to find a solution how to bring these molecules back into the muscle. And all of the solutions we try to develop, each of them has its own advantages and also its own challenges. And we are all aware of the advantages and also of the challenges. And that's why I think it's great that we actually develop these independent approaches, because it increases the chance that one of the approach will eventually make it towards the clinic. And this is the ultimate goal of all of us. I think this you all know that, and I think this everyone is maybe not so familiar with, and this is the work I do daily is looking at mice which have lamin alpha 2 deficiency. And I think there, in terms of other animal models, it's actually a huge advantage of this disease in terms of doing research with, is that in each animal, including mice, when you find mutations in the LAMA2 gene, it causes the same disease. And actually mice, we can genetically modify to have mutations in the LAMA2 gene, but there are also mice which naturally have mutations in the LAMA2 gene. So also there in mice, just by chance, you can have mice getting born which have mutations there, and then they have the very similar disease as the patients. There are actually also reports of cats or also of dogs, which just are also born with mutations in the LAMA2 gene, and then they have exactly the same disease as the human patients. And this shows us and demonstrates how important this laminin protein is, and it also helps us to understand the function of it by actually analyzing these mice. And this guy here, so both of these mice are 5-week-old. And so this is the brother of this guy here. They both have the same age, and but this guy here is much smaller than its brother, which has the same age. And so our challenge is to bring in this laminin protein, and we can do this by taking laminin 111. This is one possibility. And our approach is based on another laminin. So now we have laminin 411 coming into play. And this laminin, so all these numbers show here just that these are a little bit different versions of laminins. So this is laminin 411, and this laminin protein usually don't really found in the muscle fibers, but you found it in the blood vessels. And we all have laminin 411 in our blood vessels. That's why when we look at the muscle here, and you are familiar also, I think, with this section versions of muscles we do, and here you see like this round shape. Here are the muscle fibers. And when we look now at this laminin alpha 4 protein, we see that this is present here as this puncta, because this is found in the blood vessels, and these blood vessels are between the muscle fibers. Now we observe that in mice that don't have laminin 211, and also in all the patients that have mutations in the laminin alpha 2 gene, one finds this laminin 411 protein present at the extracellular matrix of the muscle fiber. So this demonstrates that when laminin 211 is not there, actually the muscle tries to make a solution for it and produces this laminin 411 protein. And this laminin 411 protein may actually allow that muscle fibers are even formed in the absence of laminin 211. But yeah, the muscle tries to compensate, but obviously it cannot greatly compensate, because we still have in the mice, in all the other animals, and also in the humans, we have this very severe disease. And the reason is that this laminin 211 and 411 are a little bit different in function. So the 211, which is usually there, is anchored to the muscle fibers, and the laminin 411 is hanging here in the air, so it's not really anchored to the muscle fiber. And this is the observation that is made by research, and now we try to somehow give it this missing anchor here. And for this, we designed these proteins, which we call linker proteins. And because the challenge there is, as mentioned before, is we don't want to bring into the body something that the body has never seen. And that's why we try to make use of the toolbox that is already there. And we try to take domains or parts of protein that are also found in the muscles of the patients. And for this, we take another protein, which is called agrin. So the name is not really important here, but this is also a huge protein, and researchers have studied the function of this protein. And they actually found that this protein can bind here to laminins, and it also can bind to this guy down there. And we make a mini version of this guy. And when we add it, it should actually do this. So this is the theory. Now this laminin protein is also anchored here to the muscle fiber. And there is a second problem between the two protein is that laminin 211 is usually there, can actually bind to other laminins via this arm here, we call this. And only in this case, it binds, it forms a really stable scaffold. And this guy here also one part is shorter and missing. And that's why, although it's anchored here, it forms only a very labile scaffold, which is not really stable. And we also try to give this function here back by adding another linker protein. And here we also take the toolbox from other proteins. Here also we make use of laminin alpha 1, which you have heard before. And we just take parts of this laminin alpha 1 and we make like Lego games. So we assemble this in another conformation with another protein. And this guy should then bind here and give this protein this function.
Suggestion, that's a great explanation of the principle. Can you then, just so that we don't run out of time, summarize where you're at and where you're
going? So this is So this is just this is actually what I wanted to show you. This is just the principle of the idea, and the advantage of this idea is that these guys here are small enough that they fit into these vehicles which can be used for gene therapy. And we use here the same vehicles which are used for the drug used approved for SMN, for example. And we are making versions of these vehicles where we pack in the sequence or the tool which then produces this protein. So we produce these vehicles, and what we currently do is we then inject this into the mice, and then we analyze how much this improves the muscle dystrophy of these mice.
So have you actually done this in mouse models already? Yeah. And then what type of duration or what type of response do you see from the muscles after applying this therapy?
Yeah, so we actually see that. So you have seen that these mice are very, very small. So we also say they don't thrive, so they don't really gain weight when they have the disease. And we see that the mice, when we treat them also in this case, very, very early, very soon after birth, that we see that these mice of around 4 weeks after treatment, they start to gain body weight. And they also are around 8 weeks, they're around doubled in the body weight compared to the mice which have the disease. And we also see that the muscle function, so we can measure the strength of these mice. This is much stronger when we treat them with this gene therapy.
The principle that this therapy addresses, as well as the laminin 111 and the laminin alpha 1 upregulation therapy address, is this missing link between the muscle and the matrix, this kind of disconnection that happens because laminin alpha 2 is not there. And circumventing the problem that laminin alpha 2 is too big for traditional gene therapy. So these are 3 approaches now that ingeniously circumvent that problem and reestablish the link to the matrix. By using natural kind of approaches, laminin alpha 1 as well as these linkers.
Thank you very much for giving me the opportunity to present a bit of our work. I will try to squeeze it into 10, 15 minutes, or what's the idea? So yeah. But we can always come back to things later or more privately if you want to discuss with us during the meeting. So we took a bit of a different approach. So we basically are using muscle stem cells as our therapeutic strategy, and basically all the strategies mentioned before can also be incorporated in stem cells. So the principle of it, this is also on behalf of Bram. So maybe just this is Sarah, the daughter of Bram. She has MDC1A, a LAMA2, and this is what I presented during the scientific meeting. I will not go over it all again, but I will just give you a few of the highlights to be able to understand. So I will just give you the overview of the experimental approach. That's what is the strategy we're facing. Then we're close to a safety and efficacy trial using these stem cells, and this is something I want to talk to you about it. Maybe I leave the correction out because that's more comparable as what has been discussed before. So that's not really new. And that's also with the upregulating. So I'll probably focus on the first two parts to show you what's different, what other have been done, and why we think this is a feasible strategy to move on. Well, basically, the principle is quite simple. We all have muscles. We know if we have muscle damage that our muscles are able to repair. We know if we are active and sporty that our muscles are going bigger. So the muscle is an organ which can repair itself, which can go stronger if needed. And the reason for doing this, and that was also mentioned in the first talk, is because we have stem cells. We have stem cells which can replenish the muscle. Well, there's different types of stem cells. So the satellite cells which have been mentioned, they make new muscles, but they are very difficult to culture. You cannot do them in large numbers. So we were looking if we want to use stem cells, so you can compare it a bit like a bone marrow transplantation, and you bring in stem cells into a person, then you want to have stem cells which you can culture in large numbers. Well, the stem cells we're working on are called mesangioblasts. These are the type of stem cells which I come back to in a moment why we think they're feasible. But the basic idea is that we take a muscle biopsy. From that muscle biopsy, we can culture the stem cells, so the mesangioblasts. We can culture them in huge numbers. And then if we have sufficient numbers, we can give them back to the person. And these stem cells you can inject in the bloodstream. So you can inject them in the arteries. And through the arteries, they move into your muscles, to the small blood vessels. And then these cells can move out of the blood vessels into your muscle. So this is a way of getting novel cells into your muscles, which might be able to take over the function of the existing muscle fibers. Well, if you have muscle stem cells which are healthy, then you can just culture them and put them back. But we work with a genetic disease, so your muscle stem cells, if you would put them back and just culture them, that would not be helpful. You could think about donor stem cells, and then you say, well, we don't have the problem of the genetic disease, but then you obviously can have the immune reaction. So that is why we still want to work on autologous. That's autologous means stem cells of the person themselves. And then we need to correct them. And we did some famous work on how to correct these type of mutations, and that's the technologies we can use there. But you should indeed see, we try to bring back in a person his or her own stem cells, which are healthy and do not carry the genetic defect. And we think then you will recover the genetic disease. Well, the stem cells we use, you can see them as the purple ones. They're on the blood vessels. So they're throughout your body. You can find them at multiple places. And they can proliferate. That means that we can culture them to large numbers. We can culture them into the billions. We need also to treat a person billions of stem cells. And they have a high myogenic potential, which means that they're very capable of making new muscle fibers. So they're capable of making new fibers. As I said, they can go through the blood vessels, so you can therefore put them in the bloodstream, and they go then outside to be able to repair the muscles. And they can differentiate it only as so stem cells, if you have stem cells that can become any type of cells. These stem cells are a bit more fixed. They can only make muscle type of cells, so you don't have the problem that you inject them, that you get all kinds of side effects. They just stick to becoming muscle. It has been successfully applied in dog models of Duchenne muscular dystrophy. I can show you in a moment. This is then the whole idea. So you have the stem cells, they become myocytes, they become myotubes, and then you see they have multiple cell nuclei in the whole cell. So that's typically of muscle, that you have fibers which are consisting of multiple nuclei. But these mesangioblasts can then generate new myotubes, new myofibers, but they can also fuse with the existing ones. So you have your muscles, but if you bring them in, they can fuse with the existing fibers, and that means that their content is released in the muscle fibers themselves. So in that sense, they can also repair the existing ones. So they share then the content. So you bring in an existing myofiber which has the genetic defect, you bring in a new nucleus which can produce that protein. So that means that it can also repair existing fibers. Well, this is how they've done that with the Duchenne dogs, but maybe I skip it for this talk and also to be. And This is just to show you, I showed you in the beginning, we work on a number of diseases. And the reason is not that we think we will be able to solve all those diseases, but some of the diseases allow us to make a quicker step into the clinic. And then if we show that it works for that disease, it also means that it would work for other diseases. And that's basically why we move up and down. So this is not to confuse you further, to have another disease involved, but this is where we can show that this approach leads to renunciation of muscles. And that, sorry?
Bert, there's one question is, can this be replicated for peripheral neuropathy or just for muscle regeneration?
This is for muscle regeneration. Yeah, yeah. This is for risk. Yeah. But I can show the clinical trial in a moment. I think that's helpful.
Yeah, we have only 5 minutes left. And I think this the stem cell therapy raises a lot of questions that we probably want to answer. So if you just finish with the clinical trial.
I will finish with the clinical trial. Yeah, so I will briefly, this is simple to understand that this is an affected muscle which has poor functioning mitochondria in red. The green ones are the good ones. If we bring in normal stem cells, we expect that it will shift to the normal ratio. That is just the principle. And that's typically for mitochondria, they produce your energy. Here you have only a few producing sufficient energy. Here they all produce sufficient energy. And if we bring in healthy stem cells, then we would expect a shift. And well, also there you can say, well, if your stem cells have a defect in your DNA, why can you bring them in? But we were lucky with this disease, and that's quite typical of this disease, that we have patients which have high mutation loads in their muscles. So they have 81% defective mitochondria, 3%, but they have stem cells which do not have that. So by nature, and that we don't know why, they make healthy stem cells. So for us, this is the ideal model. So we don't have to correct things. So we just can isolate those stem cells which are free of the defect which they have in the muscle. And this gives us the best opportunity to test our approach, because we don't have to manipulate, and because it's the patient's stem cells themselves, we can move easily into the clinic because we just bring cells they have already themselves. And we can see if this strategy is valid in what we want to achieve. So, and then I will finish with the clinical trial. So what we, sorry. Yeah, sorry, that is, I move a bit quick now.
The question was using stem cells, is there any worry of immune system becoming aware like with the viral vector?
No, this is autologous. So it's the stem cells of the patients themselves. Oh, sorry, I have to repeat the question. Maybe okay. It's okay, they understood, yeah. So the whole idea is that you take the patient's stem cells themselves, so then you don't get the immune response. So you cannot get the immune response. And you can also use the stem cells to bring things in. But the whole idea is, and then we take a muscle biopsy of the patient, we culture them for 50 million cells. And now we're doing a safety trial, so phase 1/2 trial, which is looking at safety. Is it safe to bring millions of stem cells in your bloodstreams without any damaging effect? And do they get to where we want them to get? So we look limitedly to where they get. So we culture the stem cells, we bring them in. The stem cells will hopefully go to the affected muscle. We do that in the underleg, just to be in control of where they go, just for the safety. That's not for eventually the clinical improvement, just to test the safety. And we check them after 1 day and after 4 weeks. And then this is then my last slide to wrap up. So after the first day, we look for safety. Did something go wrong, and do you get an adverse effect or not? We do not expect it because it's cells from your cells, so why would it be? But yeah, you can have something maybe in the bloodstream. And we label them, and we made them green. And that's to be able to discriminate them between the cells you have from your own and the ones you bring in. So we can look at the green guys in the muscle to see where they pop up, and to see how many stem cells went to the damaged muscle and how effective it is. Do we get more? And then after 4 weeks, we look again, we look at the treated muscle. Do we have more muscle fibers? And we treat one leg and we compare it to the other, which is also an advantage because we have the person himself or herself as control. So we can just treat one leg and do not treat the other. Well, we do all the procedures themselves, which means that you also get rid of biological variation you might have if you compare people. So we were lucky that we have most of our things in 2. So we have 2 legs, so we can use one leg to treat and the other leg to control for the treatment. And this trial will start in a week. So in 2 weeks, we will do it. We would have liked to present the data already, but due to COVID things, it was very difficult to plan these trials in the hospital. Yeah. And then you can say, well, this is a patient group which makes healthy stem cells. For the LAMA2 patients, we have to correct these stem cells, and there's a few other slides I have on that topic, but that's pretty much comparable of what Dwi is doing and others are doing to be able to correct cells and to be able, and we really rely also on the work of Dwi, which unfortunately she could not show in the magnitude she has worked on it, but that we join forces to help these things further, and we do it ex vivo, they try to do it in vivo, and I think these complementary approaches will eventually help us to go further. Shall I leave it here then, Carsten? Yeah.
Great, yeah. So just to give a little bit of context. So stem cells exist in any tissue of the body ready to repair that particular tissue. They're specialized to do that. And so to give you an example where this type of approach has been very successful, that's in blood disorder. So you have stem cells in the bone marrow that you can take out, culture, and actually repair out of the body and put them back into the bloodstream when they go, where they then go back to the bone marrow and make a corrected version of the cell. So that's been in the clinic and successfully done for anemia, for a type of sickle cell anemia, where the bone marrow red blood cell stem cell was taken, repaired, and put back into the bone marrow to make now normal blood cells. The challenges here, of course, are greater because, you know, muscle is a much bigger tissue that you have to get to if you want to efficiently correct all muscle cells with a stem cell approach. Stem cell has to go there and be ready to replenish the tissue and be surviving in that tissue. Yeah. And the second challenge that Bert alluded to, but that I just want to kind of bring out again, is the immunological challenge. If you take that stem cell out and you repair the LAMA2 itself, which you can, you know, maybe with a CRISPR approach, then the stem cell would make a LAMA2 that's never been seen by the body before because there was a mutation. So there you could run into an immunological problem, which probably can be managed, but that's why your approach is to use laminin-1.
Yes, also to look at LAMA1 again.
To make the stem cells make LAMA1 instead of LAMA2 to replace it. So just for a little bit of editorial background. Yeah. I'm sure that.
And it's also, if you say, well, we heard yesterday patients, sometimes what do you really like to treat? Sometimes it's also local problems. And then we might be able to tune it because we can look at where the arteries go to and then just treat specific muscles, which might be beneficial. But that's a bit further ahead, but that might be that you can be more specific on where you allocate your treatment.
But it really is truthful. It's an order of magnitude more complex than just treating the bone marrow. That's
that's
for sure.
That's for sure.
Questions?
Can this be done with other disease corrective modalities? There's a question. Can this be done with other disease corrective modalities? I don't understand the question. So
for other diseases?
Yeah. So it's this one here. It says, I think this is answering the previous question, but this can be done.
In principle, it can. The whole idea is that you bring in healthy stem cells which could recover muscle. So you could do it for more. You could even think about non-genetic muscle diseases like cancer or whatever. And that is what we also use to get funding for this research, because we're not only fixed on the rare diseases, but they can benefit from the applications which might be broader than this. And that can help you to raise the necessary means to do this.
Cool.
Yeah.
There's a question here. I'll just let you answer in chat. They're asking for a link to what you've published. So I'll let you just put a message in there for the link.
I'm happy to do so. Yeah.
I think compared to say that this is a very different approach from these stem cell clinics that you see advertised all over the world. Where I think this is very important to point out. Because those are scam operations, they're really rip-off operations where patients go and get infused with some random stem cells and
it reflects to my last slide.
Thank you. So please don't confuse them. This is a highly scientific, rational approach. It has nothing to do with
That's true. Thank you.
Whenever you say and I'm not going to be able to say right, the immunological compromise, what exactly does that mean? Like, what would that look like if someone was compromised that way after they did this
So that's something that the medical field has done now for decades and decades. It's basically dampening the immune system, directing the immune system to tolerate something that's given to the body that wasn't in the body before. That's the trick. And so the most classic example is organ transplantation. You know, when the people started to first transplant an organ, take from a different person into a person who would need the organ, like the kidney, there was initially just catastrophes. The immune system just rejected it and the patients died sometimes until the field figured out that you have to modulate the immune system to make it not attack the foreign organ and then tolerate it over time. So that's basically been highly, highly successful. I mean, there's millions of transplantations all around the world that work on that principle and work on this principle. And in gene therapy, whatever modality we do, we sometimes have to give the body a protein much smaller than an organ, a protein that the body has never seen before. So we apply the same principles of immune modulation and induction of tolerance in the long run that we've learned from the transplantation field over decades. And so it is nothing that is entirely new. It's just an application that we now use for the gene therapy. And so that's a toolbox, if you will, that medicine knows how to use.
Relative to that, could it harm the patient or would it just not work?
Yes, I mean, these therapies do come with risks, and one has to kind of carefully monitor that and tailor make it to the patient, because the patient will be immunocompromised to some degree from that, but there's nothing that you can't manage.
So the one thing I would say about this is that the basic immunologic data to know how much tolerance each individual might have for laminin-2 production is unknown. In the Duchenne world, this was learned because many of the deletions are like chunks. And so, okay, you have never seen that chunk of dystrophin, but wow, you've seen all these other chunks. They're just not working. So that ability to look in laminin-2 exactly to see fragments may be being made, and those your body may say, yeah, I know that piece, but I don't know this piece. And I think we don't understand the basic immunology of what is known in patients with LAMA2 yet.
And in approaches where you don't give a foreign protein like the LAMA1, either upregulation of protein or the linker proteins that are basically the body's own proteins just engineered to the right size, there your immune modulation is right only around the tool that you use to deliver it. So then the immune modulation is to make AAV tolerable while it's being given. And so that's only restricted time and you don't have to do it for a long time. So it really has to be tailor-made for the approach that you use.
Dr. Bonnemann, we had one question that we were asked earlier, and I thought we'd ask it while everyone's still here. The question was around about money and funding and how that is able to speed up development. And if there are, maybe you could speak a little bit about, there seems like there's three kind of like players in where funding goes. There's the science, there's the NIH, which is like almost an intermediary, then there's pharmaceutical companies that are working on development. If we could speak about how funding helps each of those three different groups and how they get funding would be interesting. And then also the next question is going to be around what else slows things down other than just money. Is there anything else families or patients can do to help to speed things up, whether it's the CMDIR, having correct information in there or other things like that?
Yeah, it's a really complicated question and probably goes beyond, and it's very nuanced on many, many levels. So if a therapy is still in the lab, have to try to prove its metal to be kind of preclinically optimized and is in an academic lab, like in Dwee's lab, she needs funding, right? Because she needs grant money, academic funding and advocacy funding to move her research in that space. If therapy is picked up by a company or budding company like Seal Therapeutics that Judith introduced, or more mature like Modalis, They also need money, but then money comes from other channels. That comes from investors who put their bet on that therapy to move forward into the clinic, and sometimes that's not enough. And then the company will need to pull in advocacy money as well, or federal money, which all these models exist to do that. But when a therapy goes towards the clinic, things get exponentially more expensive, as opposed to what happened in the lab at the bench. I invite everyone in the room to speak up for that. So probably in amounts of money that are beyond reach of a smaller advocacy, and they really need buy-in from investors and federal sources to do so. There are, and then I finish in that we talk, there are kind of alternative models that are being developed at the moment, where an investigator, maybe in partnership with an advocacy, can come to a federal and philanthropic sources and apply for funding for that particular therapy. And then the federal funding and the industry philanthropic arm will kick in and eat the costs for the toxicology, for the gene therapy production, and for instance, and the clinical development. So two of these are the Bespoke Gene Therapy Network, and we can write it down. That is a partnership between, it's an international partnership, but NIH, FDA, several industry partners, Foundation for the NIH are involved. The second program of note is the Urgent NDS program, where you can apply with any therapeutic approach. It gets all reviewed, it's all milestone driven, and where the program will then kind of pull in providers for the different things that are so expensive, like toxicology and production. And so these are just started last year and have to prove their mettle.
Yeah, so definitely from the academic point of view, I think I cannot speak on behalf of everybody, but the urgent part that Carsten brought up, I think by training and by nature of my work, my goal is to improve the efficacy. Now, the safety and the toxicology studies, not something that I've ever been trained for in my whole education. How to take from cell and mouse to non-human primate is not something that I know how to begin. I don't know how to ride it. I don't know how to buy monkeys in a layman term. So I think my goal right now is to get to the urgent stage, so that the urgent platform, so that I get help on how to translate the know-hows further.
Well, question just really quickly on sort of what families and parents and caregivers can do to help influence this. Funding, at least within the United States government's controlling Congress, are we aware of any bills, House resolutions, any government officials that are working on actively promoting and pushing more funding to the NIH that we might be able to sort of as a group kind of call and put some pressure on to move things forward and move a little bit more quickly? Yeah. I try not to look at you when I ask the question.
I'd like to comment on a little bit of both of those questions, both of the things that you guys talked about from a pharmaceutical perspective. I want to say that Modalis is really committed to this community, and we do have the money to run all of our safety trials and our phase 1 trial. So we are committed and we can do that. But that being said, Modalis is quite small as compared to a lot of the big players in the pharmaceutical field. And I personally believe that the patient voice is incredibly important, and doing things like you guys did yesterday, like the patient-focused drug form with the FDA is very important. They need to hear from you, and pharmaceutical companies need to hear from you about what you need, right? And what you're looking for, because we don't want to create drugs or solve problems that you guys don't feel are a problem, right? We want you involved. And I think that the FDA and the NIH, they need to hear from you, post about it on Facebook, go to the runs and the walks, like generate awareness for your community in any way that you can, and let people know that you want a solution to this problem. So that way the bigger companies hopefully will also hear you as well, I would hope. And I don't know of anyone particularly that you can write to, but I think being involved with the media anytime that you can to just generate awareness is good.
And there's one more point, which is where you guys all can kind of chip in big time, which is what we tried to pitch to the FDA yesterday, which is the message, we are ready for clinical trials. So that clinical trial readiness is a message that needs to be out there loud and clear. And Dr. Connolly is leading an effort, an international effort now to establish more clinical trial readiness across the field. A clinical trial readiness that should be available for everyone who wants to move a therapy toward the clinic. So it's not exclusive. It's just as a community of affected people, we are and caregivers and clinical scientists, we are ready to move something that looks highly promising into the clinic. We have the tools, the outcome measures, the ways to do that. That's enormously important to raise to the FDA, but also to financial contributors, and they have to see this field is mature, this field is ready to go, and so.
I actually want to add too that once big pharmaceutical companies are looking for numbers, perhaps a way is also to have international patient register, not only in the US. I'm from Brazil, and I actually tell all my patients to be registered in CureCMD. And right now I'm translating the website in Portuguese so it can be more friendly to these patients. But I think that has to be done worldwide, so you can have a bigger number to present to pharmaceuticals, that it has a lot of patients who are interested in that.
Yeah, the CMDIR, I think you guys hear that often. I remember when we first kind of got connected to CureCMD, we filled out the CMDIR, and it was a lot of work. There's a lot of questions, and there's also a bit of work to keep that updated. But that is the place that we use. That's the mechanism we use to share patient information to pharmaceutical companies at CureCMD. So it's very important that repository of information around patients is accurate and represents our population. And actually, we completely changed the registry in the last 2 years to make it a bit more user-friendly and a bit easier for us to manage. And so I think advocating for everyone to keep their information up to date. If you hear about a patient or another family, just telling them that this does take some time, but getting your information into this system is really important so that we can have as big a number represented when a pharmaceutical company shows interest in developing a drug. Yeah, so the question is, if you're in the registry, what do you do to keep it maintained? It's stuff at the moment self-imposed. I think we're looking into ways to have some sort of notification or reminder to go in and update. But at the moment, yeah, it's just you just got to remind yourself to make sure that your information is up to date. And on top of that, I think as it kind of connects to funding, obviously everyone here has probably raised some money for CureCMD in the past. CureCMD has a grant cycle that every 2 years we fund. So there's the amount does change. Sometimes it's $500,000, sometimes it's $250,000, and we ask for the people in this room to come and apply for a grant. And so CureCMD is spending a lot of its money that we raise to help fund the early science and the people in this room, and also paying for some of the people in the room to come to this conference to present and to share information. So there's always a balance for CureCMD when we do raise money, how to spend it. I think there's always a bias with families, like we want all the money to go towards research and drugs, but then there's also awareness and information, and not just cures, but also, quality of life improvements and things like that. So as it goes with money for CureCMD, it's a balance that the organization tries to strike, how to raise money for the science, but also how to raise money for information, awareness, funding of getting the people in the right room so this conversation can happen. Are there any other questions?
Yeah, I just want to make a comment as an academic researcher. Our technology has been licensed to some companies. The problem when you're academic researcher, as I mentioned, once it's been licensed, people think, oh, everything is fine. But the academic lab, which has come up with the idea, needs to continue to do the research to understand how it's working. And I get funded through NIH, but we compete with so many other grants, and our funding percentages are very low. And so the voice of the community to the funding, like we get funded by NIAMS or NINDS. These are, and we have program officials that if they know that there is a patient advocacy out there, they are responsive. So if you get a score that's close to being funded, they may flip it over and actually fund it. So it's important, I think, for the advocacy groups to really reach out to NIH, to those study sections that are involved in reviewing our grants, so that they understand the demand is there and we can continue the research. Otherwise, the money goes to something else, right? And if that happens, then of course our research labs sometimes move to a different different area because we don't receive the funding that we need to continue the basic research that's needed. So I think that's one thing that I think the patient population can, and the parents can help out.
Is there a way for us to know when you need us to like step up and advocate at the right time?
I think generally, contacting NIH and engaging with them all the time is an important part of that. Carsten is one of those people that can moderate that. But I've had grants that were 1 point off being funded and didn't get funded. So I mean that's how hard it is because the competition is so hot. You're looking at 8 to 10% of the grants being funded, and you're looking at each cycle maybe 100 grants being submitted, right?
You told us when you were going to submit.
I don't know if we can influence it that way.
So first of all, I'm completely hermetically sealed off the funding apparatus of NIH. I'm an NIH scientist. I'm not allowed to even talk to them, not allowed to get any money from them. But what you can do is, the program officers are the ones that kind of run the study sections and are involved in issuing RFAs, requests for applications. And so that's where the most effective apparatus is. If you get an institute to have an RFA that is dedicated for translational questions of certain science, of certain aspect, then they will issue a request for application, and people like Dean, Judith, and anyone else, or Dwi, can apply for that, and that goes into a peer-reviewed study section where only the peers have a say, no one else, and that can't be influenced in any other way. But the RFA, so the request for application, that's a really good mechanism to get money out the door for ML.
Can anyone in this room speak to how things change between America and elsewhere in the world? Like, is there some dynamic at play where there's better funding and there's a better lane in other countries, and just the fact that the research can get done there with better funding then helps everything move forward faster?
So I would say in Europe, the situation is quite similar than in the US. You have funding agency that actually support mainly also basic research. But also what actually makes me really happy in terms of patient advocacy, that they are growing also in Europe, because like some years ago, I came the first time to Cure CMD meeting 3 years ago to Chicago. And this time we never had such a meeting in Europe. And now we have also, thanks to affected families, we have a lot of patient organizations also that are growing in Europe, and then also promoting the awareness also, and also allow us to increase money sources, I would say also for Burkin also.
We did is we included the patient and patient organization in the project. So Bram is our communication advisor. He's aware of what we're doing. So we team up very closely.
Grant mechanisms that are actually requiring that.
Yeah. So that is also means that he's updated on what we need, what we are aiming for. So and he is also able to attract resources for us again. So because of the other way around, it works both ways. So I think that is quite an efficient way of keeping each other involved and connected and being able. And the climate in Europe, we have a good grant, I think, for this type of work, a few million grants, which is European money, regional money, but also the local politicians, they're very sympathetic for these type of projects. So they're very keen on proceeding this, for taking it forward. So I think creating the awareness, but also the connection is really helpful in making these things. But because not all grants are purely competitive in science, sometimes are also more political oriented. And then the sympathy factor is way more important than the scientific level. Yeah. Sorry.
Yeah, maybe I can add Oh yes, my name is Bram Verbrugge from Stichting voor SARA, but I'm also here to represent the LAMA2 Europe, because we're now working together with Impulsity, the Spanish patient organization, and the French, LAMA2 France. So I think because of that, yesterday I met someone from Turkey who is joining the LAMA2 team in Europe, and I'm talking by email with some German families. So we're trying to make it bigger and bigger, and of course also to connect with Cure CMD. And if we can have a big patient organization in Europe and connect very well with QCMD in America, yeah, I think that would be the ultimate goal.
Okay, we have like 7 minutes to use the bathroom and have coffee before the next session. So thank you all for your time.